This show me is going to focus on both osmosis and tonicity. So osmosis is movement of water through a permeable membrane. Cells, for example, as you can see with my little red cell right here, cells are permeable. They have these little channels in them called aquaporins that allows water in and out of the cell. So this is a cell in a salt solution. As you can see, my little orangey yellow dots are representing a solute, which could be any salt you can think of. And again, as you can see, it's pretty much the same salt concentration inside and outside of the cell. So we're going to have water trying to move out of the cell through these aquaporins and back in through these aquaporins. Because it's the same salt concentration, this is going to occur at the same rate. This is called an isotonic solution. Iso meaning same, and tonic representing tonicity. There is the same salt concentration, so water is moving in and out via osmosis at the exact same rate. There are two situations where this isn't occurring, and that's either hypertonic or hypotonic, and we're going to talk about both of those today. So here's our little animal cell in a hypertonic solution. This means that there is more salt in the solution than there is in the cell. This presents a problem for our little animal cell because water is always going to want to reach equilibrium. So to do this, water has to leave the cell and enter the solution through those aquaporins. By this happening, the cell is losing much needed water. So you're going to go from this healthy cell if it was in an isotonic environment, and because it's losing water, the cell is going to shrivel up. And we call this plasmolysis. Conversely, we can have a hypotonic solution, which is what we're going to focus on next. So here we have a hypotonic solution. This is an animal cell in pure water. So there's no salt in this water at all, but there is still salt in this animal cell. Again, this water is going to want to try and reach equilibrium. So in this case, the exact opposite is going to happen. Water is going to try to move into the cell until it reaches equilibrium, which it obviously can't ever do. So water is going to continue to enter the cell, and again, it presents a problem for our little cell. So we're going to go from a healthy cell this time with the addition of a ton of water, and the cell is going to become really filled with water until it eventually bursts because it can't hold anymore. So either a hypotonic or a hypertonic presents a problem for your little animal cell. Plant cells do something pretty cool that animal cells can't. So I'm going to focus on those and give you an example of both of those to hopefully drive the point home and give you something that you can visualize and see in your daily life. So here are two plant cells in two different solutions. Hopefully you can figure out which one is which by now, but if not, here's a little help. This top one is hypertonic, and this bottom one is hypotonic because it's clearly pure water. Plant cells have a cell wall. Animal cells do not. And this allows plant cells to do something kind of cool that animal cells can't. In the case of a hypertonic solution, your plant cell is going to have all of its water trying to leave the cell, which, as you might remember in our animal cell, causes plasmolysis. In our plant cell, however, we'll have the cell shrivel up, but it still has a cell wall holding it up. So while it presents a huge problem for an animal cell to lose all that water, plant cells can still hold their structure because of that cell wall. It doesn't need that internal water to hold structure. Now in our hypotonic solution, remember the animal cell got so big that it eventually burst. So our plant cell is going to fill with water just like our animal cell did. However, its cell wall has a special function. While your plant cell will fill with water and get really, really big, it's going to be pressing against this cell wall, but the cell wall is going to be pressing right back, which prevents your cell from bursting. That cell wall is essentially protecting the cell itself. So remember, while our animal cell shriveled all up in a hypertonic solution and got so big that it eventually burst in a hypotonic solution, plant cells have that extra protection from its cell wall. So think of a wilted plant. A wilted plant is a plant in a hypertonic environment. It doesn't have enough water. That plant is kind of wilted and shriveled, but the plant's still a plant. It's not a shriveled up mess. Conversely, think of a plant that you just watered. If you put that much water in surrounding an animal cell, the animal cell will burst. But a plant cell that just got watered a plant that just got watered looks very happy and very tall and not wilted at all. And that's because of that cell wall pressing that pressure in and causing the plant to be turgid. That's what this is called, when that cell wall pressure and the pressure from the cell that is in that hypotonic environment push against each other. So hopefully this was helpful and you have a little bit better of an idea of what tonicity means and how it relates to osmosis. If you have any more questions, go to your SI leader and hopefully they can help you out some more.